All right, biologists, this is cell respiration higher level from topic 8, subtopic 8.2. So cell respiration involves the oxidation and reduction of electron carriers. So we saw in the standard level that during cell respiration, we have the production of NADH. We also have the production of FADH2. These molecules are known as electron carriers because they will receive a hydrogen with a pair of electrons, and these electrons will be carried to the electron transport chain. So the energy of these electrons can be used to power the production of ATP. They are fundamental in the production of ATP in aerobic cell respiration. We have two words here for those of you who do not take chemistry. These might be new words, or at least words that you haven't heard in a while: oxidation and reduction. Let's review real quick. You remember the electron, that particle, that subatomic particle with a negative electric charge, right? Well, when you're talking about reduction, we are talking about a reduction in the electric charge, a reduction in the total electric charge. So if I had NAD plus and it turns into NADH, well, let's take a look at the electric charge. What is the electric charge here? Well, it is 1 positive. What is the electric charge here? It's 0. So I went from 1 positive to 0. Did the charge increase or decrease? It decreased, right? It went down. That means this is reduction. Why did the charge decrease? Because you got the pair of electrons that comes with this hydrogen here. Reduction is when you receive electrons. Electrons have a negative charge. So if you're receiving negative charge, you are decreasing your charge. You are reducing your charge. On the other hand, if your charge is increasing, we call this oxidation. If your charge is increasing, we call this oxidation. It's the opposite of reduction. And then we have this famous mnemonic called oil rig. Oil rig. Oxidation is losing electrons. Reduction is gaining electrons. So when you lose an electron, that's oxidation. When you gain an electron, that is reduction. Not only that, if you get an oxygen, well, that is oxidation. When you lose an electron, that's oxidation. And when you lose hydrogen, that will also be oxidation. If you lose the oxygen, that is reduction. If you get electrons, that is reduction. If you get hydrogen, that is reduction as well. So here, you might not see the electron coming, but you see the hydrogen. So this is the reduction. On the other hand, if you go the opposite way, you have oxidation. So during glycolysis, if you watch the standard level video, during glycolysis, you have the reduction of NAD plus. But during fermentation, you have the oxidation of NADH. One way is reduction, the other way is oxidation. Example, you have here pyruvate that comes from glycolysis. During fermentation in animals, we have the production of lactate. Well, look at this distinguished gentleman. Look the way it sits. Yeah, very distinguished. You have hydrogens here that you didn't have before. If you get hydrogens, that is reduction. So the pyruvate is being reduced to lactate. Now look at this guy, NADH. NADH now has a positive charge. It had charge 0, now it has positive charge. And the hydrogens are lost. This is oxidation of NADH and reduction of pyruvate. The reactions are always coupled. When someone is losing an electron, that electron has to come from somewhere. That electron has to come from somewhere. So someone is being reduced while someone else is being oxidated. These reactions are coupled every time. If you're getting an oxygen, you're getting it from someone else. If you're getting a hydrogen, you're getting it from someone else. If you're getting an electron, you're getting it from someone else. So reduction will always pair with oxidation. And this is why the electron carriers are so important, because they will take part in this whole oxidation and reduction of molecules that are derived from glucose. So pyruvate came from glucose, and all this oxidation and reduction will happen with the help of electron carriers. Phosphorylation, another word for us. Phosphorylation makes molecules less stable. Well, we talked about ATP. This guy here, this large molecule, is ADP. So here we have the A, we have one phosphate, we have another phosphate, so ADP. And then you have one ion of phosphate here. Phosphorylation means adding a phosphate. So adding a phosphate. Remember that to add this phosphate, it takes a lot of energy. Molecules with more energy are less stable, more reactive, which is the case of ATP. Now you have the A, one phosphate, two phosphate, three phosphate. This is ATP, right? So when you add the phosphate, you have a higher energy molecule, a more reactive molecule, a more energetic molecule, less stable. That means it can react real fast and release this energy so that the cell can perform work. Again, glycolysis is converted to pyruvate in the cytoplasm. We have the energy investment phase when you have 2 ATP being used to form 2 ADP. But then these 2 ADPs will be used here in the subsequent reactions to form 2 ATP. This is the energy harvest phase. So you're going to invest 2 ATP, but you're going to collect 4 ATP, right? So if you subtract the 2 that you invested, the net gain, the profit is 2 ATP. Besides, you also have electron carriers involved, right? So this NADH will be used again in the last part in the electron transport chain. So the net gain is 2 ATP and 2 NADH. Right, in the aerobic cell respiration, pyruvate is decarboxylated and oxidized and converted into acetyl compound and attached to coenzyme A to form acetyl coenzyme A in the link reaction. So the link reaction is the link between what happens in the cytoplasm, here the glycolysis, and the Krebs cycle. So we have here this micrograph, this electron microscopy image of a mitochondria. This is artificially colorized, right? But basically we have our mitochondria like this. So this space inside here, this is called the mitochondrial matrix. This is where the Krebs cycle happens. So far we had glucose making 2 pyruvate. This pyruvate will then be actively transported to the matrix. Okay, this is active transport. And then in the matrix, you have the formation of acetyl coenzyme A. You need acetyl coenzyme A for the Krebs cycle so you can continue the respiration, the mitochondria steps of aerobic respiration. So here we have these molecules. Now, do you need to know the structure of this monstrosity? No, but you need to know that this is coenzyme A, also known as CoA. And this is pyruvate. Also, you do not need to know the structure. It's just here. And then the link reaction is this. Now take a look at this guy. So the link reaction is this. Bam. Now you have this 2-carbon compound here we will call acetyl. And now you don't have just coenzyme A, you have acetyl coenzyme A. And this acetyl coenzyme A will be part of the Krebs cycle. Now you don't have pyruvate anymore, you have CO2. So when your elementary science teacher said, well, we breathe in oxygen and we breathe out carbon dioxide, well, this is the first molecule of carbon dioxide that you produce in respiration. When you consume fatty acids, when you are using fatty acids instead of glucose as a source of energy, of course, you're going to skip the glycolysis part. Fatty acids do not go through glycolysis, but they do go through reactions that will form acetyl coenzyme A from the long hydrocarbon chain. So one fatty acid can give you many acetyl coenzyme A depending on how long the fatty acid, how long the hydrocarbon tail is. But one glucose molecule will give you 2 pyruvate, which in turn gives you 2 acetyl coenzyme A molecules. Now in the mitochondrial matrix, we have the Krebs cycle. It goes by other names as well. And since biology has as many words to be memorized as a language ab initio, we're gonna learn other names for the Krebs cycle. You never know. Krebs cycle, also known as citric acid cycle, also known as tricarboxylic acid cycle. They're all the same. Where these names come from? Well, Krebs from the guy involved in finding out that, hey, we have a cycle inside our mitochondria. Citric acid, because the first reaction will give you, well, citric acid. And tricarboxylic acid, because guess what? Citric acid has 1, 2, and 3 carboxyl groups. Do you have to know every intermediate? No, you do not. So let's take this and simplify. So the pyruvate had 3 carbons, right? And from that, we make acetyl coenzyme A, and the coenzyme part that is going to be restored. The coenzyme part does not go into the cycle, so we're only going to count the acetyl, which has 2 carbons. What happens to the other carbon? It leaves in the form of CO2. We also have the production of NADH. So pyruvate will be decarboxylated. It means it will lose a carbon here, it will lose a CO2, and it will be oxidated. It was going to lose the hydrogen and the electron. So the pyruvate will lose the electrons. That means we make 1 NADH. But this is the link reaction. The Krebs cycle will start when this acetyl will join an oxaloacetate, which has 4 carbons. So the acetyl coenzyme A plus the oxaloacetate will form this 6-carbon compound that we do not need to know the name, citric acid. And through a series of steps that will give you a 5-carbon molecule, you will have the formation of CO2 and NADH. Then we have the formation of this other 4-carbon molecule with, again, the formation of CO2 and NADH. This other molecule will change again to restore oxaloacetate. And when it does that, it will not make any more carbon dioxide. You already have 4 carbons, but it's going to make, you guessed it, NADH. And this other electron carrier that we like to call FADH2. Oh yeah, oh, you almost forgot. Here in the process, we also have the formation of 1 ATP. We're lacking space here. We're gonna write ATP. This is the Krebs cycle. Now, you cannot forget that this happens twice for every glucose molecule. 1 glucose molecule will give you 2 pyruvates. That means 2 link reactions and 2 Krebs cycles. So you have the formation of 2, 4, 6 carbon dioxide, and the electrons and the hydrogens from the glucose molecule are being captured by our electron carriers. Now we're going to take these electron carriers to the electron transport chain. So the energy released by oxidation reactions, what oxidation reactions? Glycolysis, link reaction, and Krebs cycle. The energy released by the oxidation of the molecules, starting all the way back with glucose, is carried to the cristae of the mitochondria by the reduced electron carriers. What is the cristae? What are the cristae? So in the inner membrane, you're going to have proteins. We're going to have special proteins that will carry out the electron transport chain. So in order to have more inner membrane surface, so you can have more inner membrane surface, the inner membrane is folded. It has these folds here, like intestinal villi. These folds are called cristae. 1 crista, 2 cristae. So these cristae are there, so you have more surface to volume ratio of the inner membrane here. So you can have more of these proteins that carry electrons in the electron transport chain. So let's zoom in a little bit. And here we have the intermembrane space. Out here we have the cytoplasm, and here we have the mitochondria matrix. We have these 3 proteins and 2 accessory molecules that carry electrons. NADH will come here and give away the electrons. So these electrons are here full of energy, and the energy of these electrons is used to pump hydrogen from the matrix to the intermembrane space. So we're gonna pump hydrogen here, right? So the NADH will keep giving electrons to this protein, and it will pump hydrogen there. These electrons will move to the next molecule and then move to the next protein here, and then keep moving and moving again. Every time it's going to pump more hydrogen. The electron is going to lose energy. The electron is going to lose energy because this energy is being used to pump hydrogen into the intermembrane space. So the intermembrane space is getting full of hydrogen ions. It's getting full of hydrogen ions. FADH will do the same thing, but instead of starting here in the first protein, it's going to start here in the second molecule. So all these electrons are being passed around, and as they move from one molecule to the next, they lose a little bit of energy, and this energy that they lose is being used to pump hydrogen, H plus ions, to pump protons into the intermembrane space. Now we have a problem. The hydrogen doesn't want to go there for two reasons. One, all of these guys have positive electric charges, and all these positive electric charges are repelling each other. They don't want to be there squeezed in the intermembrane space. It's like the pressure is too big. They don't want to be there. They want to be away from other same charged particles. They want to be away from other positive particles. So they are pushing each other. They don't want to be there, but the electrons are forcing these hydrogen ions to be pumped there. So the pressure is building up in this intermembrane space. They don't want to be there. Also, there is a chemical gradient. So you have the electric difference, you have the chemical gradient. Here, the concentration of H plus is much bigger in the intermembrane space than in the matrix. So you have all these H pluses here that want, by diffusion, they want to go back into the matrix. They want to go back here because there is too much. There are too many hydrogen ions in the intermembrane space. The concentration is too high. The number of positive charges is too high. So there's this electrochemical gradient here across the inner membrane. There is this pressure to make the hydrogen come back. And the molecule, the protein that will let the hydrogen come back to the matrix is called ATP synthase. ATP synthase is the name of this enzyme that makes ATP using the energy of all these H pluses, all these protons, the pressure of all these protons that want so bad to get out of the inner membrane space. They want to get out so bad. They say, please let me out, I'll do anything. And then the ATP synthase says, sure, just come this way and make the machine work. You might have seen maybe a water wheel. So when you have a river, for example, flowing this way, and a farm, you might have a wheel here that spins using the energy from the water that passes here and move these pedals and move the wheel to generate work. Well, the ATP synthase works pretty much the same way, but instead of using water to move it, it's going to use protons. So the protons are going to come through the ATP synthase. They're going to come back and they're going to make the ATP synthase move and make ATP. Like lots of it. 32, 34, depends on the math you do. In real life, it would be closer to 30. And all this energy here is being used to make ATP. That is great. Now you have a second problem because all these H pluses here are going to mess with the pH of the matrix. All these H pluses here are acid. So we are making the matrix more acid than it should, which is not good because all these enzymes in the Krebs cycle, well, they are enzymes and enzymes are proteins. So they have an optimum pH. If you make the pH more acid than it should be, then the rate of reaction will be lower, and you do not want that. So you want to get rid of this H plus. That's your first problem. The second problem is, let's say that this guy here gets a pair of electrons from the transport chain. Now, another pair of electrons is coming, and this guy has another pair of electrons and more electrons and more electrons. There will be a time it has so many electrons, it cannot hold any more electrons. And then it is going to stop here. Well, but we still have this portion of the chain until this guy gets saturated with electrons. And then this guy gets saturated, and this guy, and that guy, and then no more hydrogen is being pumped, and then no more ATP is being produced, which is very bad for you. You need ATP for everything. So how can we avoid this from happening? Well, we need someone to receive the electrons. We need a guy that we call terminal acceptor. The terminal acceptor is the guy who accepts the electrons after this last protein. At the end, terminal, at the end, it will accept electrons. And this is oxygen. So all these electrons here are going to come to the oxygen molecule that you need to breathe in. So when you breathe in oxygen, this is what the oxygen is used for. The oxygen is the terminal acceptor. You need oxygen so that the electron transport chain can keep flowing. So you have oxygen, you have electrons, and you have this H plus here. Well, hydrogen, oxygen, some electrons to make some covalent bonds, and bam, you have a water molecule. So to summarize, here we have our friend, the mitochondria. We have the outer membrane, the inner membrane here with the cristae where the electron transport chain happens. We have the intermembrane space that is used to build that high concentration of H plus that then travels down through the ATP synthase by chemiosmosis. In the matrix, we have link reaction, we have Krebs cycle, we also have some ribosomes here, and we have mitochondrial DNA. So mitochondria has its own DNA, circular like a bacteria. It has ribosomes, just like a bacteria ribosome, and it has this membrane here like a bacteria, and the outer membrane is believed to be derived from a phagosome, right? So the idea of the endosymbiotic hypothesis is that bacteria, once upon a time, millions, billions of years ago, bacteria that could oxidize molecules to obtain energy were eaten, were incorporated by larger eukaryote cells, started to live there with their own DNA, with their own ribosomes, their own proteins. It doesn't mean that mitochondria are bacteria. Mitochondria cannot live without a cell, but the prevailing hypothesis is that mitochondria come from these bacteria billions of years ago. Right, everything starts with glucose in the cytoplasm. So glucose will, from one glucose, you're going to get 2 pyruvate. You're going to get 2 ATP and 2 NADH. This is called glycolysis, and it happens in the cytoplasm. Then pyruvate will be decarboxylated. That means you're going to lose CO2. From the 2 pyruvate, you lose 2 CO2. And from the 2 pyruvate, you get 2 NADH. This is called link reaction. And it happens in the mitochondria matrix. Then the acetyl coenzyme A will go into the Krebs cycle. The coenzyme A will go back for another pyruvate, but the acetyl part will be combined with oxaloacetate and go into that journey that ends up in oxaloacetate again. From the 2 acetyl coenzyme A, you get 4 CO2, 2 ATP, 6 NADH, and 2 FADH2. The Krebs cycle also happens in the mitochondria matrix. And then the electron carriers, all these guys, the electron carriers will come to the electron transport chain with the help of our friend oxygen, who is also part of the electron transport chain. You will end up with some 30 ATPs and 6 water. The electron transport chain happens in the inner membrane, the region we call cristae, right there. Right, how do we know what a mitochondria looks like? Through electron micrographs, electron microscopy imaging, and electrotomograph, which is basically taking many electron microscopy images of the same organelle so you can build a 3D model of what it looks like.